So this is a normal physiology, right? So you're maintaining your plasma glucose levels after you eat. What do? You remove all these, excess amount of glucose. And in fasting, your goal is to maintain this, glucose levels. So for the RBC mostly, and then these are just normal like a homeostasis going on, and so we'll talk about what actually happens in diabetic people, right? So diabetes type 1 versus type 2, right? So in type 1, you have antibodies to islet cells, islets of Langerhans, right, which is in your pancreas, and then these cells are damaged. Your antibodies to these cells, and then eventually will recruit, neutrophils and all that stuff, and then it will just, it's like hypersensitivity and type 4, type 2, whatever, antibody mediated, and it will, kill the cells, and you will actually have low insulin, right? So at some point, they will be not making any insulin, very minimal insulin. And what happens in type 2? Type 2, what happens is, because of the metabolic syndrome, the insulin is higher all the time, because you're eating, let's say you're eating high glycemic index foods, right, all the time. And what happens is, every single time you eat, you basically flood your plasma glucose, spikes like that. I mean, it's a normal, it's 100, you're spiking it up, and then it comes down, spiking it up, and come down. So what happens is, let me just, erase this 1 second. Let's see, there's a one big spike going up, and then eventually you need to bring down your glucose levels. Mm-hmm. So if you have multiple spikes, if the spike is much faster, is happening because of the high glycemic index foods like fructose syrup and all that stuff. So what happens? You need to have increase more insulin synthesis, right, to contain this glucose levels, right? So what happens over the time? These cells that actually, for example, these cells right here, which is skeletal muscle, adipose cells, right? Mm-hmm. And skeletal muscle, adipose, and cardiac, right? So what happens in these cells is they have a receptor, right? So insulin, again, what does insulin do? Insulin also stimulate, your liver cells, right? Liver cells, what particularly? Glucokinase, right? Increase the glucokinase synthesis. So now you're removing these, glucose to bring it down this, right? Mm-hmm. So if there's so much glucose, you will need to remove it to these cells, to these cells, to these cells, right? So you use them up, and also to the liver, and liver does its own things, and fatty acid synthesis, cholesterol synthesis, glycogen stores, whatnot, right? By the glucokinase, right? So what happens in type 2 people, they eat high glycemic food, they have metabolic syndromes, they have high BMI index. For that reason, the glucose spike is always going up, right? They're drinking a lot of soda, that's high glycemic index. So every single time you just have to make more insulin than normally the normal foods, right, you eat. So doing this over time, the insulin always spiking up, your beta cells are making insulin, and that insulin eventually, how does the insulin act on these cells? For at this level, let's say this is a cell right here. Insulin is a hormone that actually works on tyrosine kinase receptors, right? Tyrosine kinases. So then this will actually upregulate, some down pathways within the cells, and this will trigger, these responses, enzymes or whatever, glucokinase upregulated, whatnot, right? So what happens is, because of this insulin, these receptors get downregulated. Right? So these receptors get downregulated. What do we call that? Insulin resistance, right? So in type 1 people, the receptors are fine, right? But they don't have insulin to work on these. They don't have insulin, so the receptors are fine. Here, Yeah, so the insulin is not there. Here, you're actually knocking out the receptors first, and your cells are making so much insulin. At some point, these cells will, beta cells, will something called fatigue. They will fatigue, and they will stop making insulin too. So in these people, they have two problems. One is first they have receptor problem, which is tyrosine kinases, right? And second, the cell will fatigue. They will have low insulin eventually. Makes sense? So we'll have increased insulin initially, number one. That will downregulate the receptor. The cells will fatigue, and that will actually create low insulin. So they have a couple of problems that's going on versus type 1 diabetes. So the whole idea in type 1 and type 2 diabetes is the insulin is not working. Is not working, or its receptors are not there. Right? So now what happens is, for example, now we have a fasting. Let's say there's a guy who's feeding, and there's a guy who is fasting. We'll compare and contrast both people. And then let's say, for hypothetical sake, to make it simple, we can say this patient has like type 1 diabetes, let's say. Type 1 DM, right? So now, let's say this guy has, during feeding, this guy has a plasma levels, and this guy has a fasting level also, right? So during feeding, this type 1 diabetes or type 2, whatever, for hypothetical sake, we will just make it more, easier. But the thing is, both type 1 and type 2, they both work, similarly. But there's minor changes, which we'll discuss in part 3. Type 1 diabetes, let's say, this guy is eating, right? What happens? Usually normal glucose is 100, right? And now he's eating, his glucose becomes what? I don't know, maybe 400 or 500, right? Which is not normal, right? So now, in normal people, like we discussed, right, there are 3 cells here, right, that need insulin is going to work on. If there's an insulin going to feed into these capillaries, right? And they will actually tell them to do what? To put these GLUT transporters, right? The GLUT4 transporters, then it's going to start removing the glucose, right? Going to burn the glucose. So that way, in these cells, you will have glycolysis going on, right? And all of that stuff. And then in liver, the insulin here, let me just also color this red here. In the liver, you have glucokinase, right? In the liver. So insulin is going to work on here as well, right? And then upregulate the glucokinase, and also going to start removing the glucose here, right? But in the liver, you don't have GLUT4. You don't really care. The only thing it's working to increase insulin is glucokinase, right? So that is the most important thing. So now, so next thing is, in these people, you don't have insulin. If you don't have insulin, what's going to happen? Well, this 400 and 500 is just going to remain elevated, and to the point that, this body, this person is, your body is, in order for your body to be thinking like they're in the feeding state, you need insulin to be working, right? But in these people, the insulin is not working, right? So even though they are eating, there is no GLUT4 transporters being translocated because there's no insulin there to be working, and there's no glucokinase upregulator because there's no insulin. So what actually happens? Well, their body, even though they are feeding, their body thinks what?
They're starving.
Yes, the body thinks it is in the starving or fasting mode, right? Because what really tells your body that, you are actually going to be feeding is your insulin, right? That's the most important thing. And then some videos, we will discuss how does your body makes insulin. So at some point we'll discuss that. But for now, just, keeping things simple, right? So this is feeding. So now this is a problem, right? So now we'll talk about fasting. So in fasting, what happens is, again, you have, what is your goal? Your goal is to make 100 grams of, keep that 100 gram, in fasting, you're maintaining glucose, right? right? So then, what are you doing here is, so even though you're maintaining 100 grams of glucose, your RBCs are happy. They're like, okay, well, you have glucose here. We don't really care about insulin because we have something called GLUT2 transporter, which is not dependent on insulin. So we don't really care. We're gonna just, as long as there's a glucose present, I'm gonna use it to make my ATP, and I'm happy, right? So now what happens? These cells. So now what happens is your body start like doing gluconeogenesis because it's initially in the fasting state, your body is doing gluconeogenesis, and then it's making what? Glucose, right? So now your 100 gram glucose is going up to 200, and then it's going up to 300, right? The thing is, your cells, like we said, in fasting, they prefer what? They prefer using glucose more than ketones, right? In the fasting state. But in these cells right here, these 3 cells again, right? What happens again is, even though these cells don't have, they prefer using glucose because glucose is going up, right? And now what happens is they don't have any basal insulin. Basal insulin is not there. Because again, it's a ratio of glucagon to insulin, right? So initially what happens, number one, what happens in the fasting state, your glucagon is going up, right? And then it will drive the gluconeogenesis or glycogen lysis, right? So that will actually increase the glucose levels into the blood. Like, it's keep on increasing. But at some point, as the glucose is going up in the plasma, the insulin start going up in the fasting. So that is what we call basal insulin, right? That's basal insulin. So that basal insulin is also going up and making sure whenever the glucose is going to 200 and 300, your bodies start putting the GLUT4 transporters in these, even though they're on fasting. So your bodies already start putting the GLUT4 because the insulin is working on here, basal insulin is working on here. And then what happens is you start using this, excess amount of glucose and maintaining it at 100, right? What happens in these people, they don't have basal insulin. So they only have unopposed. They have unopposed glucagon, right? So what happens, their body is thinking, oh, maybe these cells, so now your cells, your heart cells, your skeletal muscle, and your adipose, right? So this heart, skeletal, adipose, so these are not getting any glucose here, right? So now these cells are thinking, maybe there is no glucose present in the plasma, right? So now, even though there is more glucose present here, so they will start, using ketones, right? So your body is doing gluconeogenesis. Remember this process again? So first thing is you're maintaining the glucose levels 100, and then now your 100 levels going up because you cannot use it because you don't have insulin to use it, right? And what happens is this glycogen is breaking down, and then you starting to use you're doing gluconeogenesis, right? But the thing is, you cannot, even though you're doing gluconeogenesis, you're not using this glucose. So the glucose is going up, and now you kind of push your body into a hyperglycemic state, right? And when you have a hyperglycemic state, what does your body make? Insulin, right? You make insulin. But in these people, what happens? You don't have insulin. So now you're kind of stuck with the hyperglycemia, right? And then at this point in time, what will happen is your body wants to again spare the proteins, right? Remember the spare the proteins we talked about last lecture? So spare the proteins, what happens is your fatty acids breakdown is start trending up. Gluconeogenesis is going, and your fatty acids breakdown is going up. This is your fatty acids breakdown going up. So now you're making ketones, right? Usually, in normal people, so like, let's see, normal people, your glucose stays 100 and you start making ketones to spare the proteins, right? But in these people, their glucose levels are also going up, right? Because they are not able to use any glucose. Even though cells prefer glucose, but they can't, not all cells can, let me see here, not all cells can use glucose. Make sense? Mm-hmm. So if they cannot use glucose, then this is like, even though some cells are using it, but to really bring it down to 100, you cannot use it. So because of the insulin is basal, even the small amount of basal insulin is not there, right? For that reason, this is going up, and now your body is not able to use glucose at all, and it's going to start sparing the protein, right? And the ketone is going to go up, right? But now what happens is the ketones here, however, not dependent on insulin, so they can use the ketones, right? So that's what's happening here. So to put it all together, is